morning. Did everyone have a good evening? So, do I get to ask questions? No. Okay. So you're taking no questions. So I encourage everyone to ask Dr. Sadowski questions in between the sessions. That was a great presentation. And where Dr. Sadowski started and referred to during her talk, I agree with wholeheartedly. There are multiple evidence-based, proven interventions to improve function of individuals with motor weakness or paralysis from a neurologic event that are available right now. And so our two talks being juxtaposed are quite appropriate because I'm going to talk about a therapy that is not available right now, a therapy that we're developing, a therapy that we hope will help, but as Dr. Sadowski started, will be of zero value to an individual unless they are actively pursuing rehab strategies, activity-based therapy on a daily basis. Only insane clinicians in Dallas would put people to bed for 3 months and study the impact on the human body, but that's the type of stuff we do in Dallas. But what happened to those individuals in 3 weeks was profound, and what happens in even over 48, 72, 96 hours of inactivity is pretty profound. And so there is a use it or lose it phenomenon when it comes to all of us, but more so in individuals who've had a nervous system insult. And so as I tell my patients, and some of you are unfortunate enough to be in that category and have heard this, you don't get better by going to therapy, you get better by doing therapy. So we get asked all the time, my insurance company isn't approving 2, 3, 4 times a week physical therapy, to which I say, I don't care. You can still do therapy even on your own, and the therapist is there to train you on how to do that and do that effectively. So even if we all win the lottery tonight and we take a group trip to Maui, you still do your therapy every day on the beach in Maui. So with that as a backdrop, I wanted to take a few minutes to give an update on where we are relative to the stem cell trial. How many people here in the room are familiar with the fact that we are pursuing a stem cell trial? So 1 or 2 of you, got it. All right, so this has got a fancy title, the study to investigate safety of the transplantation of human glial-restricted progenitor cells. Well, what the heck is that? I'm going to explain it. First, as a backdrop, people have asked, why does it take so long? What do we have to do to actually get to a study? And shouldn't I just get on a plane and go to one of the many advertised stem cell clinics around the world and get injected with whatever they're injecting? And I'm not 100% sure what they're injecting. But here's the reason it's a process. So first, if you're going to do this in a rigorous fashion and you're going to determine that an intervention is safe and effective, there's a lot of what's called preclinical work. If I'm going to inject something into a human being, you would like to know what it is in general. We've heard the stories now of Zantac that's contaminated with different things that are cancer-causing and other generic agents. If you haven't heard this, it's in the news over the last few weeks that the FDA has shut down the importation of certain generics because of contamination during the process, and this is why we have an FDA to make sure that what we put into the human body is as safe as can be and has a chance of working. So there's a lot of preclinical development that goes on to sort out whatever the therapy is, and in this case it's cells. So then you do preclinical testing. You do as much as you can in a dish or in an animal to ensure that whatever therapy you're looking at does what you intend it to do and doesn't do anything surprising. Then you go to the FDA and you apply for what's called an IND, an investigational new drug. You submit an investigational new drug application. And this is my favorite part of the government. So they don't say yes, they just don't say no. So they don't. So you send this application and you wait 60 days. And if they don't say no, you can go ahead and move forward. You get a letter ultimately saying, the IND's been granted, but it's not a stamp of approval. It's just they're not telling you don't do that. And then you're allowed to go into human subjects. You are not allowed in the United States to do research with an experimental agent, something that has not been FDA approved, unless you have an IND. So we can't just, if I have a bottle of some sort of humoral salts in my lab that I think really is great, and I give it to you, I go to jail because I have to have FDA approval to study in a human any type of pharmaceutical or therapeutic agent, which brings into question a lot of what happens in the world of supplements that advertise a medicinal benefit. So all those fast-talking lawyers at the end of the ads that say, this is not approved to treat or cure any medical condition, that's to avoid the FDA coming down and saying, you're advertising something that hasn't been tested in a rigorous fashion. And then you get to move into what's called a phase 1 trial. And there are multiple stages of trials, phase 1, 2, 3, and 4. So phase 1 is a small number of subjects purely to understand safety. If I give somebody this agent, will they grow a second head? Surprisingly, none of the mice did, but will something random happen to these individuals? So it's purely about safety. And then you move to phase 2 and phase 3, which are meant to balance understanding both the safety and efficacy of an agent, because the FDA was founded not just to ensure our safety, but also to protect ourselves from snake oil salesmen who will sell things that are perfectly benign, but have no chance of helping you whatsoever, and take your money happily, but really have nothing to show that it makes any difference whatsoever. And both of those are actually dangerous. Things that cause harm and things that are completely unefficacious pose different types of harm. So let's put this in the context of stem cells and go back to the preclinical development. So we've known about the power of stem cells, embryonic stem cells, for 50 years. And the power of these cells is that they can turn into any other cell in the human body. And you can use them in a variety of ways. So if you take a human stem cell and you put it in a dish and you walk away and you give it some food and a nice little rec room to play in, and you leave and you walk back, what you're going to find a couple weeks later is a lot of different cells. You're going to find some heart cells, you're going to find some muscle cells, you're going to find a little tooth growing, you're going to find a little bit of neurons, and they just divide into all the different things they can divide to. The challenge in stem cell therapy, the reason it's taken 50 years to get to a place where we're actually talking about therapeutic interventions, is scientists had to determine how can you control the fate of a stem cell, because as I like to joke, and some of you have heard, you don't really want a tooth in your spinal cord. And so if we just take random stem cells, put them in your spinal cord and come back later, there's going to be all sorts of things there. And so the idea, and one of the preclinical requirements that the FDA placed on this research, was we had to prove, scientists had to prove, that the cells would generate just the type of cell you want, and they wouldn't generate anything else. Well, how do you prove a negative? So you have to grow these cells, and you have to put them in animals, and you have to follow those animals for a really long period of time. And you have to do it over and over and over and over and over again. And you have to say, listen, we tried this 1,000 times, and we haven't seen any teeth. All we see are the cells we want. And so proving a negative is quite difficult, but that's been one of the challenges. So in the title of the slide, where we talked about glial restricted precursor cells, it's a specific type of cell that is derived from an embryonic stem cell that can only turn into one of two cell types, something called an astrocyte and something called an oligodendrocyte. That's what the glial restricted precursor cell is. So the cells that we're proposing to put into human beings are determined to only do that and not form a tooth or a muscle or liver or anything else. And it's those oligodendrocytes that we hope will remyelinate a spinal cord. And in fact, that's what we've seen in animals. And so what was done in this setting for preclinical development was to take certain cells, these NEP cells, and differentiate them into glial-restricted precursor cells, and then to show that they only form the astrocytes, or what are called the oligoprogenitor cells, which then mature into oligodendrocytes and make myelin. So we know from a variety of studies, both human-based and animal-based, that oligoprogenitor cells are the most hardy cells for growing new myelin. And it turns out that human beings have these cells, particularly in the brain. A lot less is known about the spinal cord. My suspicion is there are far fewer in the spinal cord, but that is not an evidence-based fact. We know that they exist actually to a great degree in the brain. What's interesting about them in the brain is often they will go to a site of injury, start to remyelinate, and do a haphazard job. They don't finish the job, and there's work trying to figure out why that happens. But what we know is that the cells will migrate to an area where there's a lack of myelin. They will stretch out their arms. They will wrap their arms around the axons and create new myelin, such that signal that was once blocked from getting from point A to point B because that axon didn't have any coding will now transmit appropriately and the signal will get through. And so the preclinical testing of the cells for remyelination have involved a variety of steps. We've taken animal models of demyelination. So for example, you can take a mouse that doesn't have myelin that is destined to die because of its lack of myelin. The signals can't transmit from point A to point B. And we've put these cells, the glial restricted precursor cells, into the mice and looking for remyelination. And during that process, what we found was the cells grow myelin and the mice survive. And so the brains develop, the nervous system develops, and you rescue this mouse from a fatal outcome. But also in these models, we've transplanted the cells to make sure nothing surprising happened. Did the cells that we thought would only become oligoprogenitor cells, did they turn into brain tumors? Did they turn into cysts? And at least in the animal models, that has not been seen. And so all of this went into the preclinical testing relative to these cells that we're talking about today. So you take all of that data, the preclinical development data, the preclinical testing data, and you put together this application called the Investigational New Drug Application, and it includes all the things that are listed there. Do we think preclinically the cells work? Did we see any safety outcomes, toxicology data, injecting the cells into animal after animal after animal to see if anything bad happens to heart, liver, lung, kidney, anything that would be surprising? And then, especially for cell-based therapy, there's a lot of oversight of manufacturing. So the FDA has to certify that where you produce the cells is following very strict procedures such that batch to batch are the same, such that batch A doesn't have a contaminant when batch B was okay, and it's really there to protect us. And so there's a lot of manufacturing data that goes into it. And then you have to have a proposal for how you would study the cells. So you present in your application to the FDA, we plan to do a human study, and here is how we're going to do the human study. This is where the FDA enters into a conversation with researchers and clinicians, where they can write back and they say, we don't like the fact that you are including patients who are left-handed or whatever the case may be. We think that's dangerous. Whatever the issue is, and you enter into a negotiation with the FDA on what would be the most appropriate trial design for a first-in-human intervention. And there are, as you can imagine, lots of discussions. If you've never given an agent to a human being before, ever, and somebody's going to be first on planet Earth, then the FDA is quite interested in sorting out, well, how did you decide that that person was going to be first on planet Earth, and are you going to put them at excessive risk? And so you enter into this discussion relative to how to conduct the trial, who you're going to include, what are the endpoints, how are we going to know if the safe and efficacy is there, and then ultimately, what is your oversight and safety reporting system? So that's backwards. So there are a lot of challenges when we entered into this process. So the first was, who should we enroll? Should we enroll individuals who are within the first 30 days of their transverse myelitis, their spinal cord inflammation? Should we enroll people who are 90 days to 6 months? Should we enroll people who are 50 years after their event? What would be the most appropriate patient from a scientific perspective, balancing safety and balancing the desire to see efficacy to enroll in a trial, and frankly, the pragmatics? For people who are within the first 30 days of their event, is it likely that they're all going to be able to transfer to a site in Dallas and take part in a study? So we had to balance a lot of different features when picking the time frame to enroll. How do you deliver the cells? And this is an area of much controversy. You can go a lot of places and get an IV infusion of something labeled as a stem cell. When we have done this over and over and over again in labs and in humans, 99.999% of those cells get cleared by the lungs and the liver. They never make it to the heart, they never make it to the brain or the spinal cord. And so intravenous delivery of stem cells turns out to be an extremely inefficient way to get stem cells to the site of nervous system injury. And so what's left? Well, you can put it in spinal fluid or you can go directly into the spinal cord, but those become more invasive procedures? And if you are going to do those procedures, what are the unique considerations? What do you have to take into account? How do you monitor safety? Well, depending on the patient population you enroll is going to dictate looking for these safety events. Do we just call you up on the phone every couple weeks and say, hey, how's it going? Do we do a couple MRIs? What do we do to monitor the safety? How do you monitor efficacy? How do we know if it's working or not? So, and this relates to who we enroll. Let's say we were to enroll somebody who was within 60 days of their event, and I give them stem cells, and then they go see Dr. Sadowski at Kennedy Krieger, and they work their tail off, their activity-based therapy, and they get better in 9 months. Was it the stem cells, or was it the rehab? And so I could very easily design a study that proves aspirin cures transverse myelitis by giving everyone in my acute setting aspirin, sending them to KKI, watch them get better and say, see, aspirin cured transverse myelitis, but it didn't. Christina cured transverse myelitis. And so you have to monitor for efficacy in a way where you'll trust the results. And then how long should we follow patients? Is it enough to follow a year? From a safety perspective, you might consider lifelong follow-up. And how do I get people to sign a consent form that says, for the rest of your life, you're coming to the big D. Either I die or you die, and that's what ends the study. But that's not a very appealing way. So we have to come up with a time frame for follow-up. So where are we relative to this stem cell trial? And most of you are aware the preclinical development was completed, the preclinical testing was completed, the IND was granted, and the phase 1 study is now open for screening. And so what's listed here and what's on the screenshot here is the website. We are doing this in partnership and could not do it without what was once the Transverse Myelitis Association and is now the Siegel Rare Neuroimmune Association. And on their website is the link to go in and go through a screening protocol. So every trial has inclusion-exclusion criteria, and I'll show you what the screening looks like. So once you go to that website, there's a study details, and then there's a tab that says how to participate. You can click on that, and it's going to take you to a link. That thing that says AIS SW Med is basically a survey. It's a series of questions that we developed based on our inclusion-exclusion criteria to save you a trip to Dallas. Because when you answer these questions, we can tell you based on the answers, is there just no chance you're going to qualify? Don't waste your money on a plane because we already know you're not going to qualify. Or, yeah, it's looking like you might qualify. Let's get a little more information and make sure people are only taking trips to see us if they have a high probability of passing the inclusion-exclusion criteria. So what are the types of questions that are going to be asked? So here's a little bit of the screenshots. Are you between the ages of 18 and 70? That's an inclusion criteria. So this is not targeted for pediatric patients. And then I get asked all the time, why do I endorse ageism and have a cutoff of 70? I do not endorse ageism. It is standard to have an upper age for any trial relative to the things we're going to do to an individual, because once you sign an informed consent, here's a little known fact. So let's say you come to the clinic and you pass the screening, you sign the informed consent for the stem cell trial. And on your way home, I've done nothing to you, but on your way home, you have a car accident. That's reported as an adverse event in the trial. From the moment you sign consent, everything that happens to you gets reported to the FDA. And so as we get older, our risks of other medical events go up. So there is usually a cap. Frankly, usually it's in the 50s. We expanded it to 70. To avoid the other medical things that can happen to us and avoid it being ascribed to the therapy when in fact it had nothing to do with the therapy. The next question is, are you less than 10 years from your onset? That is an inclusion criteria. We needed to pick a range of who we would include, and the range is more than 1 year out, less than 10. So between 1 and 10 years, and I can discuss why. Are you able to walk or stand? If you are ambulatory, you are not a candidate for the trial. The decision was to take individuals who are not walking, and let me explain why. Our route of administration is a direct surgical injection into the spinal cord. We are going to be taking patients to the operating room. Our surgeons are going to open up the spine. There's a device we use. A needle goes into the spinal cord, and the cells are infused so that there will be zero question of did the cells get in or not. We know the cells are getting in. And in discussions with the FDA, the preference was to start with individuals who are not walking, such that the surgery itself wouldn't pose a risk to the nervous system. Now, to be clear, this device has been used in studies in other ways, and we have not seen spinal cord injury by injecting into the spinal cord yet. But for this condition, transverse myelitis, for a first in human, the decision was to start with individuals who were not walking and were not standing. We're screening for history of blood clots because we're concerned during the perioperative stage, around the time of surgery, as to whether or not somebody can develop clots. And then there's a list of other questions here. So if you can't have an MRI because of a pacemaker, you can't be part of the study, because part of the safety evaluation is doing serial MRIs. Because one of the things we're looking for, what's one of the unique things to this study different than any other stem cell study? Well, we're putting in these cells into the spinal cord. What if, just like when somebody transplants a kidney, you get a host response to the kidney? You've heard of rejecting an organ. This is essentially an organ transplant. I'm going to be very upset, and so will everyone in the room, if the injected stem cells induce transverse myelitis, because it's an organ transplant. Could the immune system respond to the cells that we're putting in? So everybody who's part of the trial will go on medication to prevent rejection for about 6 months, be on medication to prevent rejection. And in studies, we think that is plenty for the body to accept these cells. But we're doing MRIs to make sure that we don't induce inflammation in the spinal cord. And then we're screening for other neurologic diseases that could impact the outcomes in the study. So I won't go through all of these. They're on the website. But when you hit that submit button, it goes to my senior nurse coordinator, myself, Jan Cameron Watts. These are reviewed. And if you answer in a way where you meet the inclusion criteria, she is in contact with you to get more information so we can start going through the screening. So the study is going to include 9 adults. This is a phase 1 study, so it is only targeting safety. Are we looking for efficacy? Would I be thrilled if I saw efficacy? Sure, absolutely. But the trial design is to prove that both the procedure and the cells are safe in a population of patients with transverse myelitis. Individuals enrolling will be non-ambulatory. They will not have evidence of recurrent, a condition that leads to recurrent disease, such as neuromyelitis optica, and I can explain why for the first round we did that. I explained the direct injection. It's 9 months of very intense follow-up, so you have to be able to get to Dallas monthly over those 9 months, and then it will be 10 years of follow-up, not all in person. Some of them are by phone and by video conference, but with episodic checks. You will have to come back to Dallas in the first couple years a couple times for checks, but we're going to be like Big Brother. We're just going to pop up and jump out of bushes and say, how you doing, over 10 years. But that's our long-term follow-up. So it's worth noting a couple things. So number one is this is a long time coming. We have been literally working on this for about 15 years to get to this point where we can get to humans. The second thing to recognize is there are multiple groups, companies, other organizations working on stem cell therapies for different diseases. There was no way anybody was going to invest in transverse myelitis for a stem cell study without this community existing, without having an engaged patient population and family population, and without this community and donors community in Dallas funding the trial. This is not an NIH-funded trial. This is not a company-sponsored trial. This is a donor-funded trial. And without that, it does not happen. It will not happen. This is actually a pretty unique thing in medicine in terms of people coming together and really pushing for their population to get served. We're always trying to attract big pharma to invest in rare disease communities, but it takes a partnership, and this trial is an example of it. The other part of this is I'm extremely lucky to work with probably the most dedicated, we'll take KK on any time, team of professionals. KKI is pretty dedicated. But these are an incredible group of people at Southwestern. You know many of the folks here who are on the screen, and you don't know many of the folks who are behind the scenes, but this really takes a village. I'm the one standing up here talking and presenting, but none of this happens. The team for the stem cell trial is over a dozen individuals at UT Southwestern who are spending immense amount of time nights, weekends, traveling, doing other things to make sure every piece is put into place. And so I'm very appreciative for the team at UT and all the work they do to get this done. And so with that, I'll end with 2 more pictures of my girls, because it's better than ending with a picture of me. And I'm happy to take 1 minute's worth of questions. Yeah. Yeah, so for preclinical development and preclinical testing, it was about a decade worth of work. The IND and the regulatory process, and let me be clear, not because of the FDA, but because of the work we had to do, was probably about 18 months. Once it went to the FDA, they were very quick. We got responses in 45 days, 60, I mean, they were very efficient, and I give them credit. It was a extremely collegial, very supportive, very engaged, very thoughtful conversation on how this should be done. After we got the IND, which we've had for over a year now, we have found that there were lots of things on the regulatory side, hoops to jump through. Let me give you an example. So as part of the application, we certified that our surgeon who's doing the surgery, a colleague of mine who I trained with at Hopkins, who's a spectacular spinal surgeon, had to do 2 things. Had to go through an animal training course for the surgery, because it's a very unique surgery, and it was developed by a couple neurosurgeons, so he had to go and do this animal training course, which was fine, and we set it up, and he did it, and he passed. But for the first human surgery, he had to be assisted by one of the developers, one of the 2 folks who are certified on this device in the country. And we were waiting for a surgery to pop up elsewhere, for him to get on a plane, go fly, scrub in and do it with them and get the final check on the box. But those studies weren't enrolling. So we literally waited for months and months and months and months and months, and finally decided this just isn't working. We need to bring one of the other surgeons to us. Well, they don't have a medical license for Texas. So we had to apply and get what is called a special exemption, a fast track. And so we had to get a surgeon, a colleague of ours who's been wonderful. We give a lot of credit to Nick Boulis at Emory, who was the developer of the infusion pump. And he went through all the steps to get a license in Texas. And now he's going to fly to Texas and do first surgery at UT Southwestern with Dr. Bagley together, and then Dr. Bagley will do all the rest from there. So there have been a lot of steps to go through in order to get us to this point. And we're still putting the final frosting on the cake, but we're open for screening so that when that final frosting's there, we're ready just to go with the first patient. Yes?
I have 2 questions. So I saw on the survey, on the survey there was an egg allergy question, just out of curiosity. What was that about?
We feed everybody eggs in Dallas, and it's just one of those things. I wanted, is there a barbecue allergy? But they said, no, we got to go eggs. Everybody, while they're on the immunosuppressant agents, if we're in flu season, have to be able to get the flu vaccine because of the immunosuppression agents. And so if you have an egg allergy, it's one of the things that would take you out because you won't be able to get the flu vaccine. So.
Okay, and then my second question is, will there be now I think about it, I kind of answered it myself, but will there be a new recruitment for the phase 2 trial?
Yeah, so let's talk about where do we go from here. So phase 1 is safety. The conversations, it's a great question. The conversations have been going on for a long time of where else would we turn. And I'll give you the list of potential phase 2 or next studies. So we are very interested, extremely interested, in moving very quickly into individuals who can stand and walk some, but not the way they want. So one of the places we want to go is to expand on the exclusion criteria, who can get in from an ambulatory status. We have an interest in going into pediatrics. We have an interest in going out beyond 10 years and not having the 10-year cap. We have an interest in going into individuals who have higher lesions, going up high into the neck, which we're not doing in the beginning. We're starting lower down neck and shoulders and mid-back range for the lesion. So we have multiple places we could go. That will depend purely on the data we get from this trial in terms of safety and frankly, the investment that somebody out there is willing to make in how many of these trials could we do next. So. Yes, and the microphone is coming to you.
Is there a reason you didn't get NIH funding?
Yeah, it's an interesting question. So funding for clinical interventional trials only comes up through the NIH in very sporadic ways. The NIH does a lot of funding of preclinical development. They do a lot of funding for preclinical validation in the basic science. They do a lot of funding for mid-sized trials, so the phase 2. And those grants are really hard for those phase 2. There's something called the NeuroNext program, which has been spectacular, but those really are for drugs that have passed phase 1. So phase 1 is a tough spot for NIH funding.
If you have a large lesion that is really large, how do you inject in different places in the spine or just one?
Yeah, it's going to be a series of 10 injections distributed over a lesion. We have a maximum range we can cover. So it's 3 to 4 levels of the spinal cord, because what we have to do, so it turns out that injecting a spinal cord is really hard for several reasons. So number one is once the needle makes contact with the spinal cord, there's a layer of tissue on the spinal cord that as I push the needle, the spinal cord just starts to indent. And then it kind of like pops up a little bit onto the needle. And so getting your target in terms of where you want to be in the spinal cord is really tricky, and that's where this device comes in. Because the other issue was every time your heart beats, the spinal cord moves. Well, that's awkward as we're trying to precisely inject cells into the cord. And then it turns out that you guys under anesthesia don't just want to stop breathing for a couple minutes just to make our lives easier. So the whole chest is moving. So the device for the infusion was literally designed and had to get to the point about how long it took. The device we used to inject that Dr. Boulis developed had to get independent FDA approval, separate from the trials. So it had to go through its own regulatory process in order to get approved. What happens is the surgeon, when we open up the back and we get to the spine, it's what's called a floating derrick. It attaches, there's a screw that attaches it to the back of the spine. So as you breathe and move, it moves with you. And then the cannula floats down, and as the heart beats, it moves with the heartbeat. And then it very gently goes in. And then it turns out if you inject if I just go squirt, the cells shear in the needle. They die in the needle. So you have to do it under very low pressure and very slowly. Otherwise, so when we look at cell-based infusions, like when we put them on an infusion pump or we hang them from a bag, a ton of cells just shear in the needle as they go through. And so you have to design these systems to do very gentle pushing of literally little droplets of cells into the cord. And then what's impressive is the cells migrate. So once they're in, they start moving around. And they can go for a centimeter in each direction. They can cover a fair amount of territory. So we can do up to 10 injections up and down the cord as part of this trial. So you don't have to have a tiny lesion to get in. Yeah.
One more thing.
Yes.
With the first phase, you said they're going to be followed for 10 years. Do you have to wait 10 years to start phase 2, phase 3?
No, so we only need the last patient to get to month 9 before the data can be submitted and we talk about phase 2. And so what we agreed to, and this is an example where the FDA engaged in a very reasonable conversation. They said, listen, we want long-term safety, but let's agree to a short-term amount where there's a lot of risk, and we all agreed around 9 months of follow-up, and then they'll accept that data to consider where we go from there. But in return, we said, but we agree we will follow folks for 10 years in case something does pop up, we can report back. So. One last question. Chitra's giving me an evil eye.
Hi. So you mentioned that people with recurrent disease like NMOSD and MOG would not be part of this trial. But if there is success, is this something that could be eventually translated to that? And you know, a lot of the people have optic nerve damage. Is there a problem, I guess, with the fact that you would give this to them and then the disease would turn right around and do damage over what you just did again.
Yeah, so we have every intention for translating this to individuals with aquaporin 4 anti-MOG mediated disease, neuromyelitis optica. There's no reason that this wouldn't be helpful to at least some patients. And in terms of the optic nerve, we are very keen to inject these cells into optic nerve. The reproducible surgical techniques hadn't been developed, and for a variety of reasons, we started with spinal cord, but it is definitely on the target to go after optic nerve, 100%.